one person. Say, like, how can you do an experiment with one person? I say, I demonstrate it to you in one person. They give me anybody, right? So, and nobody would put money against me and say, who's going to put money against, I'm going to give sugar to, just bring him anybody you want from anywhere in the world, and I give them sugar, and they're going to have insulin go up, right?
So hello and welcome to the Cheeky Science Show, where in this video I had the absolute pleasure to interview Professor Valter Longo, Director of the Longevity Institute at the University of Southern California, as well as the Program on Longevity and Cancer at the Institute on Molecular Oncology in Milan. Valter Longo, well known for designing the fasting mimicking diet after making a series of remarkable discoveries in mice and then in humans that indicate that specific diets can activate cellular self-repair mechanisms and promote rejuvenation in multiple organisms to optimize health. Here we chat more about his research work and his future plans. So hi Valter, thank you very much for joining me today and welcome to the Cheeky Science Show. It's an absolute pleasure to have you with me.
My pleasure, my pleasure.
Thank you. So your research focuses on understanding how diet composition and diet timing and the underpinning mechanisms can be used to help further understand longevity and help to treat different diseases. And I guess you summarize a lot of this information in your really excellent book, The Longevity Diet, which I recommend for anyone that they should definitely read that book. And one of the kind of quotes that I'm probably going to quote wrong from the book is that you mention how because we all eat foods, a lot of us think we know that we often think we're diet experts and we know what the best thing to eat is. But actually in your book, you use a very logical approach and you use what you call the 5 pillars of longevity to come up with your idea of an ideal longevity diet. And so I was wondering if you'd be able to explain what these 5 different pillars are and how you use that to come up with your longevity diet. I realize that's quite a big question, but it's...
Yeah, no, thanks. So I always thought that it was strange that in a courtroom, we will use all these different pillars, if you will, right, to convict someone beyond a reasonable doubt, but in science, in something so fundamental for human health, I mean, we all recognize nutrition can completely change whether somebody is going to make it to 50 or 100. And so, and I thought how strange that most people use epidemiology, for example, to make a decision, right, about food. And so of course I thought, well, what is really important, right? After spending 30 years working with some of the best in the world on nutrition, I thought epidemiology, so the study of population, is definitely important. So that's one big pillar, right? So what happens to lots of people, whether they eat this way or that way, with lots of proteins, low protein, how do they perform? And then studies of clinical studies I thought were very important, kind of like, we've been talking about the high fat, low fat for cardiovascular disease for a long time. And then you have studies like the Astruc study that looks at thousands of people and they're either put on a low fat diet or a high olive oil diet. And, within 5 years, you're starting to see the difference, right? So I thought that's a really nice study that brings the clinical studies to, no, it's probably not bad to have fat if that fat is olive oil. And if you have, if you have beginning having cardiovascular disease. And then, basic science focus on longevity, So what happens with a mouse if they have a high fat diet or low fat diet or high protein? And not just mice, but what if you look at all kinds of organisms? What is the role of amino acids and proteins in making an organism live longer or shorter? So that's also very important. And then centenarians, So what about if we have all these and we put them together, we come up with something. But then it would be nice to see that lots of people that live a long life use something similar or something that makes sense, right? So if you say low protein diet seems to be associated with benefits in all these different pillars, and then you look at centenarians and none of them have a low protein diet, then you have to wonder, that's strange. Why don't they? You know, if it's so positive. But sure enough, you see them having most of them have low protein diet. It's not all of them. And then the fifth pillar is complex systems. You know, so I thought that, you know, the reductionist approach is also very important. Sometimes you don't have data. So one of the things I always mention, you know, there is some data, but not easy to interpret. So is it good to go running all the time? Is it good to run 100 miles a week, right? And some people say yes, and then, well, what about the opposite? Is it good to not run at all, right? So if you look at a car or a plane, I think most people recognize that any system that is complicated, complex, you know, like a plane or the space shuttle, you can keep it there and not use it, but you cannot also use it all the time, right? So nobody is going to want to buy a car with 300,000 miles on it. Why not? So you could say, well, it's been very, very used all the time. Well, almost guaranteed that car is going to have parts that are about to break apart, you know? So yeah, so those are the pillars.
Thank you. Yeah, no, and I really like the way that you use the 5 pillars, as I think it is, as you say, a very logical way of using the different pieces of evidence and pulling them all together. But correct me if I'm wrong, but I think probably the basic research pillar is probably the largest pillar we have at the moment in terms of evidence. And so, I mean, what would you consider are the strongest pieces of evidence we have supporting, let's say, low protein or high fat and dietary components for the longevity diets that you mentioned?
I think it's, I think it's pretty even, right? So the longevity diet, if you think about the everyday diet, there's a lot of, you know, epidemiological data supporting either the safety of, let's say, a low protein diet or with a certain composition, say 0.8 grams per kilogram per day, or the efficacy in that diet making people live longer, you know. So there's our studies and several Harvard studies and just on and on. I think if you put lots of epidemiological studies together, you come up with, you're never going to come up with something that is perfect, that doesn't have any studies against it, but you come up with a lot of support for it. So for example, I was very happy to see recently, I always talked about, you know, vegan diets being potentially dangerous for not because they cannot be done, but because they're hard to do correctly. And it was interesting that a new paper came out and showed that epidemiological data showed that vegans were 2.5 times more likely to have fractures, hip fractures, I think it was, than carnivores. But pescatarians did not. In fact, in most cases, there was a trend for pescatarians to be lower than the carnivores, right? So yeah, that is the sort of the common denominator that I look for. I mean, if you look at the pescatarian diet, of a, you know, low protein pescatarian, lots of vegetable, not so much fruit, you know, it's really a very particular type of diet. You don't see too many things against it, right? And that's what I look for. And same thing for, let's say, 12 hours of time restricted eating versus 16 hours. If you look at 16 hours, you find a lot of negatives, right? If you don't eat for 16 hours a day, lots of negatives. If you don't eat for 12 hours a day, I could never find a single negative study. And that's really what I focus on. You know, is there like this sort of jewel of interventions that you can never find something that anybody says wrong, something wrong about them, right? So I've never heard anybody saying, you know, I discourage the pescatarian diet because, you know, there is not enough proteins. Like, you know, fish, we all recognize it's got a lot of proteins, a lot of essential amino acids, very different from the vegan, right? So yeah, so that's really what I was looking for. I was looking for things that are so solid, it's just almost like impossible to take them apart. You could always find something better eventually, but I thought it's just very unlikely that you're going to find something much better anytime soon, you know.
That's good to hear because I pretty much mainly follow a pescatarian diet, so like, forgot about that. But I think I mean, I'm probably biased because most of my research is basic research, like the fundamental, like biochemical level. And I guess what I love about your research and the research of others is that these different studies have not only identified different dietary compositions that, like, have been shown to help extend health span, but also underpin like the biochemical components. For example, there's been work that's shown how following these diets or having low protein diets reduce the activity of components such as mTOR and reduce IGF-1 levels. And so I was just wondering if you could explain a bit or elaborate on these so-called longevity signaling pathways and what your understanding of them are.
Yes, so most people to this day, and I would say lots of epidemiologists, they just look at the math, right? They just say, well, you know, this is what it is. I have no idea why. I just know that people that eat this way, they do better than people that eat that way. And I think that, you know, we need to move to hypothesis-driven, recommendations. And why? Well, because, yeah, you need to put it all together. And I think that the nutrition response genes tell a story that is much, much, much clearer than the one that you get by saying, I don't know why, just lots of protein seems to be doing good. And because at a certain point, and this is why the FDA usually doesn't allow you to go forward unless you have some good degree of mechanisms in animal studies, right? It's going to say, well, you think you know, but you don't really know. And, you know, what if you were wrong about this, you know? So once you have the mechanisms, once you say, well, I know this drug lowers cholesterol, and I know that cholesterol is involved in atheroma formations and you know, atherosclerosis, et cetera, et cetera. Well, yeah, okay, now you have a story, and that the role of that drug is much more believable. And yeah, I know that high cholesterol is associated with higher cardiovascular disease. You put it all together, and yeah. So the genes, the nutrient response genes, then tell the story because alone they can extend the lifespan of all kinds of organisms, right? So if you say taurosis kinase if you say amino acids activate IGF-1 and also activate taurosis kinase independently of IGF-1. And this extends longevity, and this shortens longevity or lifespan. Well, then we already know that just by blocking by deleting or by inhibiting taurosis kinase, we can extend longevity. So it's much harder to argue against that, right? Say, well, that alone does it. We forget that you can keep the diet the same, and yeah, so they become very, very, very important. And I think the signal, the nutrition signaling genes are very important, but even more important is what I call, you know, 3 billion years of R&D, which is evolution, right? And I teach my students to look at yeast, for example. Yeast can live in the fast lane for a few days, or it can live for 17 days in stationary phase in water, or it can live for a couple of years as a spore, right? So now imagine if you wanted to go from A to C, let's say a couple of days to 2 years, right? So it's a hundreds of fold difference. You could go crazy with all this reasoning, right? Or you could say, hey, wait a minute, I think the organism already knows, right? I think already has a program in there that allows it to go from 2 days to 2 years. It's like, that's crazy, right? So why does the queen bee live for, you know, orders of magnitude more than the worker bee, right? Well, you could spend all your life trying to figure out the nutrient signaling pathways because they eat different food, and then they have a different developmental program. Or you could say, let me just activate the queen bee program and switch to this, you know, incredible lifespan. So yeah, I think that part is even more important. I mean, understanding the signaling genes is key, but even more important, understanding the program, the longevity programs. That's really at the center of the fasting and the restrictions and lots of the things that we publish in the past almost 30 years.
Exactly, and as you kind of already just alluded to, one of the great ways of understanding like the underlying mechanisms is that you can then see how different interventions have a similar impact. And so obviously you just mentioned there fasting and how it's not just about the diet's composition, but also diet timing can also in a similar way activate or repress these different signaling pathways. And so you coined the fasting mimicking diet. And so I was wondering if you could explain about how you came to create this diet and how it relates to fasting, and also going back to activating these longevity programs.
Yeah, so the fasting mimicking diet, people sometimes think, just wake up in the morning and come up with this and just become my favorite project. But in fact, as I was a student of Roy Walford back in 1992, and Roy was this guru of nutrition and longevity, right? That's all he spent his life trying to figure out, how to make people live longer with nutrition. And I realized that, what he was doing, calorie restriction. So what if you take an organism and you take about 20-30% of the calories away? And I realized that had obviously incredible power, but incredible problems associated with it. Right? And so then I spent the next almost 30 years trying to figure out how can you get that incredible power, and without the problems. And making it feasible to people. But then I started working with bacteria and yeast that were starving, and I saw that you could completely starve a bacteria or a yeast and they live very long. And I thought, this is really strange, right? It's very counterintuitive. It's like, why will any organism live so long just in complete absence of nutrients? But then I started thinking, what if we just did it very quickly, or quickly for a few days, and then allow the system to go back to the normal food, And that worked very well. And there initially was just water-only fasting for, let's say, 5 days every couple of weeks, or 4 days every couple of weeks. And then eventually went to fasting mimicking diet in mice as little as once a month for 5 days. And in people now, we think, it could be as little as once every 3 months. And why is a fasting mimicking diet? Well, we started doing a cancer clinical trial at USC Norris Cancer Center, and this had water-only fasting, and none of the patients wanted to do water-only fasting, and we thought, of course, our naive group of scientists and oncologists thought, of course, people are going to do water-only fasting. They have cancer. They're very motivated, right? This was the reason. And it doesn't work like that, right? So even with cancer, people say, I'm not convinced by this. I don't want to do it. It's too hard. So then we went back to the National Cancer Institute and the National Institute on Aging and asked to, develop the fasting mimicking diet for cancer patients. This was developed for cancer patients. And then realized that, if you have a dietitian and the fasting mimicking diet, then you can get a very high compliance. Yeah.
Cool. No, thank you for explaining that. So you mentioned clinical trials, and I was just wondering, obviously, I guess one big question is how safe would implementing fasting mimicking diets be, for the rest of your life? And I was just wondering, what is the longest trial that you've done so far, or are in the process of doing, to see how many times FMD can be done, and looked at more long-term safety consequences?
Yeah, so we are about to publish a paper that also includes a survey of thousands of patients or doctors, I think 400 doctors that follow thousands of patients. And the good news, which is kind of surprising, we really seen rarely even people ending up in the hospital, right? And now, once it gets to thousands of people, you expect that, some people by chance will end up with grade 3, 4 side effects, but we really are not seeing that. So that's the biggest evidence, I think, coming from all the people that are doing it out there. And some people have been doing it for years. And so far, I mean, you usually hear, I think the company is also obliged to report it, right? So to the FDA, so should somebody have severe side effects. So far we haven't seen it. That doesn't mean that there couldn't be some, but we haven't seen it. So I would say that seems to be very safe. And it was also designed to be that. So it wasn't designed, lots of times we get people to say, why don't you have less sugar? Why don't you have less starches? Why don't you have, less calories, etc. And part of the development of the fasting making diet for everybody, not for cancer patients, but for everyone, was to get a little bit less of an effect and more safety. Thinking of, what if everybody did this? Let's say between age 20 and age 70, the burden is to, if you want to take a healthy person, and you're working on aging, right? So no matter what we come up with, if you intervene on a sick person, there is a certain degree of tolerance for side effects, right? So if you have cancer, you have Alzheimer, yes, somebody could die even because of the therapy, but there's nothing you can do. But if somebody's healthy, right, then you have a different problem, a different burden, and you have to make sure almost nobody ever dies, right? Or even almost nobody ever has severe side effects, and that's a very difficult, that's a very high burden, and but that's what we've been focusing on, making it so that we rarely see problems with it.
Exactly. And I mean, as you mentioned earlier, one of the reasons that you created the fasting mimicking diet is to kind of avoid some of the more deleterious effects of doing water-only fasting, which could lead to malnourishment. And one thing that I find quite interesting, and you had a paper I think came out in 2019, which is how fasting can influence the gut microbiome. And obviously the gut microbiome feeds off like fiber that we consume in our diets. And so sometimes in my head, the thought of doing a water-only fasting makes me think, oh, what's going on with the gut microbiome? Have they got no food? And so obviously you showed in that study, I believe it was looking at how FMD could alleviate symptoms of inflammatory bowel syndrome. And so I was just wondering, like, to what extent do you think some of the benefits of FMD are being mediated by the gut microbiome, and what part do you think they play in the whole process?
Yeah, so the original idea, because Walford was, and lots of people that I work with were always fans of either vegan diets or pescatarian diet, and these longevity areas around the world, the Okinawa. So I thought from the very beginning, I don't want to do a fasting mimicking diet with any ingredient. I want to do a fasting mimicking diet with the ingredients that the longest lived people in the world eat all the time. And you could have argued with me and say, that makes no sense, because you're just giving it to them for 5 days once in a while. Why do you do it with use meat, use red meat, use lard, right? Use animal products. Yeah. But I thought, no, I think eventually we're going to see benefits from using the right ingredients. And, that paper was one of the first ones showing that water-only fasting actually made the gut of mice more leaky, right? And it makes sense, right? Because the mice, after not having food for a while, they start breaking down lots of cells that they don't need as much, right? So if you have just water, you can break down the gut and the leakiness increases. And but with the fasting mimicking diet, which contains a lot of vegetable, a lot of prebiotic, we didn't see that, right? So there's probably enough food in the gut, plus this feeding of this lactobacillus, bifidobacteria, et cetera, it was probably sufficient that we saw this big expansion in these protective microbes. And we don't know, but I mean, there's lots of papers that are not ours suggesting that, yes, if you go from whatever 15% to 60% protective bacteria species, then you can be expected to have benefits for inflammatory bowel disease or general inflammation of the gut. So now we have, a number of clinical trials that are looking at exactly that. Stanford is actually now taking the lead in doing the study. So we'll see what happens. But now we're sort of combining this idea of the fasting with the idea of feeding the microbes, right? So fasting while feeding, and I think that's very important. And also the post-fasting feeding is important. So what fed during the fast, but it was also fed after the fast that is probably driving, the microbes one way or another. So if you eat a steak, this could be killing a lot of the effects of the 5 days of the vegan diet, right? So so that's why we also think that at least 24 hours past the FMD should be lots of vegetables, maybe a little bit of fish, and that's it. And make like cereals, a little bit of fish, and lots of vegetables.
I'm glad you mentioned that, because that was actually my next question, was to talk about the refeeding process as the last stage after the 5 days or so of the fasting mimicking diet. And the reason I wanted to ask you about it was, obviously, what I research in a lab is cellular senescence. And I think it's thought that obviously the fasting stage, the 5 days of fasting, might help to clear potentially senescent cells. And I was going to ask you if you had any strong evidence to support that the senescent cells are being cleared. But then more on from that, the mentioning how there's currently a bit of controversy over the therapeutic use of senolytics, and how some concerns are that senolytics just clear the cells without reactivating stem cells to regenerate tissue. And so I was wondering if you think maybe therefore the fasting mimicking diet with the refeeding process as well may potentially be more superior in terms of having both the clearance of senescent cells, but also the activation of stem cells, and what evidence there is for that.
Yeah, I think the main thing is what I was saying earlier, right? About billions of years of R&D, right? So the biohacking, I think, is fine as long as very, very aware of what's always been there for the purpose of something. And we are starting to suspect that when you starve any organism, from bacteria all the way to humans, when you starve any organism, and it makes a lot of sense if you think about it, and not that everything that makes sense is true, but so you starve an organism, the organism is forced to shrink essentially, and in the shrinking process, it could do two things. It could eat anything, right, of itself. It's got to eat some of itself. It's got to, consume less energy. So it has to get rid of some energy consuming components, cells mostly, but it also has to eat itself. And so in this process of eating itself, it could pick anything, or it could go pick the worst things that there are, right, and kill those, right? And you think, is it possible after 3 billion years that an organism, whether it's a bacteria or a human being, in that moment that is shrinking itself and it's getting rid of billions and billions of cells, is gonna go, or at least shrinking billions and billions of cells, it's just gonna not care what it picks, right? I think it's extremely unlikely. It's much more likely that, like in development, when we develop, there is a lot of apoptosis, and apoptosis is looking for bad components and good components, right? So we already know we have that system, right? The human body and the mouse, they can pick and choose what's good, what's bad. And so now you're shrinking, and now food comes around, and you have enough food, enough proteins, enough sugars to regrow. And so now you're regrowing. Well, what are you going to do? You could pick any stem cell, any cell out there, or you could say, okay, let me pick the best one, right? And let me use the best one as a template to start over. And let me not just use the best cell. Let me use what I already know from embryonic development, right? Let me just use a program that has already been there from the beginning. Why not? You know, the genetic material is in there. The epigenetic information are there also. Why don't you use that? You know how to use it, right? And that's exactly what we see, right? So you see in the expansion, you look at the embryonic development genes and they're all light up, right? And these are not usually seen in adults at all, right? So and now how can you see nano again, OCT4 and all of this just lighting up in all kinds of organs, right? So I mean, it's hard to demonstrate these things all the way, but we're now seeing in the pancreas, we see in the liver, we see it in the somewhat in the brain, we see it. We see it in so many, in the gut. So now you start getting the picture. It's probably part of this evolved program to get rid of the damaged components, turn on the cellular and stem cell factors that can then rebuild, right? So yeah, so that's really the focus is the and in the process, of course, if you have senescent cells, that's bad, right? So that's something that the system will recognize as bad. Now we've been working on it for a while, and we're about to start a clinical trial on that, and so we're looking at all of that, and we'll see what happens. But hopefully we'll see a reduction of a senolytic effect of fasting mimicking diet.
Yeah, you're saying that really well, and I'm very excited to see that research. So another thing I wanted to ask you about was epigenetic changes that might be occurring during fasting or fasting mimicking diets, and the potential. I think you saw in one study, I think it was in mice, that even after doing a couple of months of the FMDs, you saw that there was like a kind of memory of the process, such that levels of like IGF-1 still remains low. And I was wondering if you've done any studies in humans where you've maybe looked at different epigenetic marks or even have interest in doing like an epigenetic age for patients that have undergone FMD to see how that might be correlating with like the biological age and whether FMD can have an impact on that.
Yes. So we are about to publish in collaboration with Morgan Levine at Yale about the biological age algorithm that she uses, which is based on, in our case, 7 markers, blood markers that are associated with a particular chance of dying in the next however many years. So we're about to publish on that. And then we are about to start soon enough with Steve Orbit. We're about to start, looking at, we're looking both at the people that are genetically, the Ecuadorians that we've been following, that have, mutation in the growth hormone receptor, and the fasting mimicking diet, looking at the epigenetic clock. And we'll see what happens in those studies. But we're close to starting both. Yeah.
Oh, cool. No, that's really exciting. I'll be on the lookout for that as well. So I guess, another question that I was interested is going back to some of your earlier work and using yeast as a model organism. And one thing as well from my own interest is that my PhD project focuses on the protein P53. And I might be wrong, but I'm pretty certain that yeast don't have the homologue of P53. And so I was wondering, do you think there's any involvement of P53 in any of the effects of fasting or FMDs or diet composition, and whether in yeast there's like some alternative mechanism that they just don't need this protein, or what are your thoughts on it?
No, P53 is definitely very central in the fasting effects in mammals. So we published several papers, particularly in cancer, but probably involved in both the cancer progression, but also the cancer incidence, and maybe the aging process itself. P53 may very well be also involved in what I was saying earlier, I wouldn't be surprised if one of the genes that are involved in deciding who gets killed and who does not is p53, right? So we see it as very central. Yeast doesn't have it. Yeast has other genes and pathways that may be precursors to p53 and the tumor suppressors. But I think, I like to call it molecular strategy. I think the molecular strategy is very similar. So yeast doesn't have insulin, doesn't have IGF-1. It's a unicellular eukaryote. It does not need insulin and IGF-1. It does not have an endocrine system. So so I think p53, there are molecular strategies that are very similar in yeast, and it just happen to not yet probably need the sophistication of p53.
And so with today, with, I guess, different model organisms being more available, so things like zebrafish and axolotls and all sorts of different model organisms now, do you still feel like there's a place for yeast? What are your thoughts on that?
We don't do almost anything if it doesn't start from yeast, right? So, every clinical trial we've ever done, everything we do usually starts with yeast. So I think that the beauty, is the fundamental property of an organism, right? So the starvation response and all our cancer work is 100% based on our yeast work. It started because of yeast. It started because of seeing that the starvation causes these transcription factors being elevated, but if you introduce a RAS-Val19 mutation in yeast, this effect went away, right? And that was it. That was our whole big operation in our many clinical trials on what's called differential stress resistance. And so I think that that's a mistake not to have a simple model organism nearby and just ask. It's almost like somebody they're very wise that you can ask questions to. How do you think this works? Is there a way to distinguish all cancer cells from all normal cells? And I think the yeast is probably it's so simple that it just gives you a real answer. It doesn't give you in any complex organism you have a million answers. Like, it could be this, it could be that, it could be a million different things. And yeast is just, that's it, By the end of it, it's like, okay, I know how it works. It's this, right? That's, so of course, worms and flies and different organism, I think model organism have a different place, but yeast is definitely, and not surprisingly, so many Nobel Prizes, right, were given for working yeast, or bacteria, including the recent ones, right? The moment you think that some of these organisms are no longer valuable, and then you get CRISPR, Cas9, and you're like, okay, well, here we go again, another study done in very, very simple organisms, so.
Very good answer. So talking about research questions, to you, what do you think are the biggest unanswered research questions we have at the moment? Or to phrase it a better way, what research questions are you most interested in for the future?
Well, we're very interested in, now that we are seeing this, reactivation of embryonic developmental programs, we're very interested in that, right? So how do you guide that? Is there a way to intervene? Is there a way to, control them more? Can you do it in the brain, right? Can you do something like that in the brain? Maybe, we could take, the mouse pancreas and damage it where it makes no more insulin. Then we start with the cycles of the FMD, and we see the pancreas, the beta cells starting to be regenerated in this beautiful sort of program. But can you actually do this in a much tougher system like the brain? And, could you do it when the brain is damaged in a mouse, let's say Alzheimer or Parkinson? And could you do it in a person? Now we have a clinical trial in Italy running with Alzheimer's disease and fasting mimicking diet. So this is very interesting, right? So is it possible that, we don't even need, there is like sort of like a black box effect where, it just works. And then we could take our time, figuring out how it works. But it could also be that, no, the black box effect is not there. We need to figure out exactly how it works. But that's going to need some, kind of like we see for cancer, right? In cancer, we've never been able to, I always say I've never been able to cure any mice with fasting alone, right? Or fasting making that alone. It's always like fasting plus or plus, right? So the last one, for example, with hormone therapy, is fasting plus estrogen receptor blocker plus palbociclib, right? And the only reason we could do that is by the molecular understanding of how the cancer cells is wired or rewired once it becomes resistant, right? So I think the future, we're looking at black box effect, but also as we get more and more sophisticated with molecular biology, but also omics, computational biology. If you start putting all these omics together and maybe using artificial intelligence, and can you now start gaining more of an understanding of the programs that are involved in making an organism younger.
And speaking of different omics technologies, what are your thoughts on precision medicine, or I guess in this case, precision nutrition, and what kind of like biomarkers or data do you think we'll need to really get to that stage? Because you mentioned in your book how you should like kind of eat like your ancestors, and how for certain reasons you might be allergic to different foods. And I was just wondering, what information do we need to understand why someone might be allergic to a certain food and should stay away from that, or you should eat this is best for you for the future?
Yeah, so I think we need both precise medicine and unprecise medicine, right? Okay. Everybody likes to go to the precise, and in cancer we see that example, right? So I hit the CDK4/6 pathway with palbociclib, but then I have the unprecise medicine of fasting that in that case hit insulin, IGF-1, and leptin. All three. So then the precise medicine is necessary. Why? Somebody from Japan that's been, lactose intolerant for hundreds of years, families, if they're gonna move to the United States and start drinking a lot of milk, they're not guaranteed, but they probably have a high chance of becoming lactose intolerant. So if you have celiac disease and you're just loaded with bread and pasta, you're gonna have a problem, right? So I think we can recognize that there are precision medicine is very, very important, and but at the same time we also have to recognize that unprecise medicine as important. So it's like what we call evolutionary medicine. Looking at, I always say, bring me a person to whom I can give sugar that they will not have an insulin response, right? So you could do an experiment of one person, right, and get it right. If it was just like, I could show you that in response to sugar, insulin is going to go up. Say, give me anybody, I'll show you. One person, say, like, how can you do an experiment with one person? I say, I demonstrated to you in one person. They give me anybody. Right, so and nobody would put money against me and say, who's going to put money against? I'm going to give sugar to. Just bring him anybody you want from anywhere in the world, and I give them sugar, and they're going to have insulin go up, right? And so that's the unprecise medicine I'm talking about, with fasting, and so there are some things that are just going to be very, very similar to almost anybody you can pick in the world. Some people do not respond with insulin to sugar, right? So but there's probably 1 in 100,000 or something like that. So the great majority of people, the unprecise medicine is as important. So yeah, I think we're gonna have to move forward with both of these. Saying what's common between a bacteria and a human, right, on one side, and then what's only common to you, and that you are celiac, and you have this mutation, and you just happen to not or have your immune system attack the gluten component of the food.
Well, I think that's a really great way to kind of summarize our conversation. It's been absolutely fascinating to talk to you, and I hope, I'm sure many of my viewers will have found it fascinating as well. So I just really appreciate the time you've taken today to talk to me.
Well, thank you. You're very welcome.
Great. Thank you very much. So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.